Some epithelial cells can be called neuroepithelium. For example, taste buds and olfactory epithelium, which you can see in this image. Olfactory epithelium is concerned with the sense of smell and is fundamentally nervous tissue. Olfactory epithelium is a pseudostratified columnar type of epithelium made up of 3 cell types. I'll describe this epithelium in more detail in my lecture on the respiratory system, but the olfactory receptor cells have slightly paler nuclei than the other cell types and are bipolar neurons and are the critical cells of the epithelium. They extend the full thickness of the epithelium and their axons travel to the olfactory lobes of the brain, which is only a very short distance away. These olfactory neurons are very unusual in that they can be replaced by new neurons even in the adult. And this is pretty important because the cell bodies of these neurons are very exposed. Taste buds are also another example of neuroepithelium. This is an image of a piece of mesentery removed from an animal and spread out over a glass slide, then stained with hematoxylin and eosin. I'll focus on more features of this spread when I describe connective tissue, but look at these long tubular-like profiles. They're actually blood capillaries. Now look at these capillaries at this higher magnification. Here is a loop and anastomosis of a very small blood capillary or venule. Note how thin the wall of this vessel is, lined by endothelium. Endothelium is a simple squamous epithelium with elongated or flattened nuclei. It's the special name given to the epithelial lining of blood vessels. Remember earlier on when I was showing you the epithelial lining of a small artery and a small vein with underlying layers of smooth muscle cells in the section of the salivary gland? And I warned you not to confuse it with stratified cuboidal epithelium lining a duct. These are profiles of sweat glands in the skin. Myoepithelial cells can be seen wrapping around the secretory cells. Look very carefully. They have the shapes of multi-armed starfish with their arms orientated along the tubular gland. So in cross sections, their arms are visible as red wedges at the outer edge of the gland. These are strongly eosinophilic due to the abundance of actin and myosin. Longitudinal or oblique sections of these myoepithelial cells show that the arms are elongated. Myoepithelial cells, like secretory cells, are ectodermal in origin and are part of the epithelium of the sweat gland. The term often used to label linings of serous body cavities is mesothelium. This epithelium may be squamous or cuboidal, depending on the location within the body. In this image of the outer lining of the colon, the mesothelium consists of squamous cells. As with endothelium, this specific terminology is not based on any morphological or functional significance. And here is a buccal mucosa scraping. Gently scraping the inside of the cheek with a cotton swab detaches cells from the surface of the stratified squamous non-keratinized epithelium of the oral cavity or the cheek. These cells are large and flat with small rounded nuclei. Here you see them face on. In this orientation, squamous cells look very different from the dark, narrow lines you see mostly in cross sections. Scrapings such as these have two major uses. One is in pap smears. Cells taken from the cervix in this manner are examined to detect cancerous and precancerous transformations. Indeed, these cells have been stained with a Pap stain, especially useful for observing abnormal cells. The other modern use is to obtain tissue in a non-invasive way for DNA sequencing. For obvious reasons, geneticists sample the cells of the inner cheek instead of the cervix. It is convenient that the surface cells of non-keratinized epithelia retain their nuclei.